All right, welcome everyone. Thank you for giving me the opportunity to present my master's thesis research today. The title of this research is the development of a quantitative optical coherence elastography probe for preterm birth prediction. Before I start, I'd just like to thank my supervisors, Dr. Alireza Mowla and Associate Professor Brendan Kennedy. Doesn't want to. There you go. I think it's working again. All right. So quickly, a quick agenda of what I'll be going through in my talk today. So I'll start with a clinical problem identification, then go into optical coherence elastography as a technique, the design and implementation of a new probe that I developed over the course of the thesis, some signal processing that was developed in order to produce elastography results, then a demonstration of that probe and validation using phantoms before doing some concluding statements. So first, clinical problem identification. So this research is based on preterm birth. So the normal age of a pregnancy is 40 weeks gestation, and preterm birth is defined as birth before 37 weeks of gestation. So the preterm birth rate globally is about 11.1%, with low-income countries having a higher rate than that, but even higher-income countries having a rate still fairly comparable, actually. However, preterm birth rates have been decreasing over the last few decades, as you can see sort of on the figure on the right. So specific to Australia, the preterm birth rate in 2017 was an average of 8.7%. I mean, there are still sort of large variations where Indigenous communities have a preterm birth rate of 14.2% and remote communities 13.5%, which is quite high. The reason that this is significant is because these rates amount to 15 million preterm infants per year, leading to over 1 million infant deaths. So in the developing world, preterm birth is the second leading cause of child death. Meanwhile, in the developed world, preterm birth is the largest cause of child death under the age of 5. So additionally, past sort of childhood, preterm birth still has higher risks of adverse health outcomes, such as cerebral palsy, intellectual impairment, chronic lung disease, and vision and hearing loss. And this is due to the fact that there hasn't been sufficient time for the organs to develop through gestation. So this has a significant impact on the health system and the community. So for preterm babies in Australia, the average length of stay is 7 days compared to 3 days for babies born at term, as well as there's a 10 times higher cost over the first year of life for preterm babies compared to those born at term. So the mechanics of the cervix plays an important role over the course of pregnancy. So at the start of pregnancy, the mechanics is firm and acts as a load-bearing structure for the fetus, allowing for development. However, going through gestation, there's a softening of the cervix before there's anatomical changes, allowing the baby to enter the birth canal and be born. This is broken up into roughly 2 stages, namely cervical softening and cervical ripening. So cervical softening is earlier in gestation, where there's remodeling of the extracellular matrix, causing sort of decreased stiffness of the tissue. Near the end, there's ripening, where there's inflammation of the tissue and causing anatomical changes and allowing the baby to enter the birth canal. So cervical insufficiency is a term that is used to describe premature softening of cervical tissue relative to the gestational age, and is linked to incidence of preterm birth. So it is stated that current interventions such as the use of corticosteroids could alone save 370,000 lives annually. However, this is dependent on the ability to identify women at high risk of giving preterm birth and therefore giving those interventions. So I think something's happened to the thing again, sorry. Thank you. So the current gold standard to measure risk of preterm birth is that of cervical length measurement, where by transvaginal ultrasound, they'll measure the length between the internal os of the cervix and the external os. The problem, however, with this technique is that it's got a low sensitivity and specificity of 37.3% sensitivity and 92.2% specificity. So this kind of relies on the fact that if you have a short cervix and during the ripening stage there is going to be changes to the anatomical structure, and if the cervix is already short, then the baby will enter the birth canal prematurely. However, there is suggestion that we could do preterm birth prediction in the cervical softening stage rather than relying on the anatomy, rather rely on the sort of stiffness and biomechanics. This was demonstrated using ultrasound elastography, which shows improved specificity and sensitivity of 78.8% compared to the 26.3% for traditional cervical length measurements. The problem, however, with ultrasound elastography, it's got low resolution and low accuracy for those particular regions. A way to address this is through optical coherence elastography. So this is a high sensitivity elastographic technique and also provides the ability for compact implementation due to being an optical imaging technique. So elastography, what is it actually? So this produces images based on, rather than the optical properties, the mechanical stiffness of tissue, and allows for a quantitative image that you can see on the right-hand side there of a soft bulk with a stiff inclusion embedded besides it. So optical coherence elastography is based on the imaging of optical coherence tomography, which if you've had an eye scan is what they're imaging your eye with. The optical coherence elastography has a resolution between 5 to 50 micrometers at the expense of a lower imaging depth of 2 millimeters. So quantitative elastography requires two components. First, you need a known mechanical load, which you can calculate the stress applied to the tissue, as well as you need a measurement of the deformation to classify the strain, which is combined to give a quantitative property of elasticity. So the research aim of this thesis is to develop and demonstrate a low-cost, compact, quantitative optical coherence elastography probe towards eventual application in vivo for tissue measurements for preterm birth prediction. So the tissue imaging to look at the deformation will be done using a forward-facing OCT fiber probe, and the stress to allow for quantitative measurements will be with a low-cost load cell force transducer that's embedded into the probe. So this will be integrated into a handheld small diameter probe to allow it for in vivo tissue measurements. So how does OCT actually work in terms of physics? So it utilizes the backscattering of light to form an image. So this is similar to ultrasound. However, the problem with OCT is that the time delay between the signal is actually in femtoseconds, making it impossible to detect via direct electronic means. However, if you use a Michelson interferometer and use a spectrometer in the detection arm, you can infer the backscratching of light from here. So here we see a broadband light source that's split into two parts. You have a reference arm and a sample arm. So they'll get back reflection from both the reference arm and sample arm, which are then combined before being separated by diffraction grading and then onto a spectrometer, which you can detect the intensity of light. So the measured interference that you get shows spectral modulation, as you can see here. And through the Wiener-Kinich theorem, what you can do is you can correlate the frequency of the spectral modulation to the distance within the sample that scatterer is coming from. So this produces what is called an A-scan, or axial intensity profile, which you can further scan to produce two-dimensional and three-dimensional images using OCT. So into the development of the probe over the course of this thesis. So they had a few different components, developing the handheld probe, the optics, the strength sensor, which are all combined in an overall probe integration, as well as tissue mimicking phantoms to develop and test the probe, as well as a scanning protocol on how to use it. So the actual handheld probe. So the aim was to create a compact and ergonomic probe that was suitable for in vivo measurements. So the design was done using a computer-aided design software, SolidWorks, and is shown on the right-hand side there. So the actual fabrication was using a high-resolution stereolithography technique, which provided 50 micrometer resolution for our prints. So the results is I manufactured two probes, one with a gray pro resin and a clear resin, which are mechanically rigid and, importantly, biocompatible, which is important for use with biological tissue. So the probe tip was 2.4 millimeters in diameter, and that was an optimization of it being small enough to be allowed in vivo, but large enough that you're not causing tissue damage, such as with a small diameter needle. This is also designed to be lightweight and ergonomic, allowing for a few different hand styles. So you can use what's called a power grip if you're applying large amounts of forces on stiff tissue, or also a tripod grip if you need to sort of fine control of the probe. So going into the probe optics that are going to image the sort of mechanical properties of the tissue, the aim was to create a miniature OCT fiber probe with high sensitivity. So we did in-house testing at the Optical and Biomedical Engineering Lab at UWA, as well as beam simulations using MATLAB. So after this, we sort of ordered in some 1D common path fiber probes. And the reason we use common path is that the reference reflection that you use for OCT shares much of the same sort of beam path as the reference reflection. So this compensates for beam dispersion or polarizations that could otherwise affect your technique. And we used an optical adhesive to encapsulate and align that optical fiber in the probe. So the results is we had a probe with a beam waist of 10.3 micrometers by 9.4 micrometers, allowing for high resolution imaging, and importantly 50 decibel common path signal from the end of the probe. We determined the imaging depth of the probe to be 600 micrometers, which is where the signal to noise ratio was in an acceptable region above 1. So next, incorporating the strength sensor so we can allow for quantitative measures of elasticity. So we needed a strength sensor that was high sensitivity and had a high dynamic range and capable of in vivo. So previous probes used a compliant layer at the end of the probe. However, this provided significant challenges for in vivo implementation if you're trying to insert a compliant layer into the body. So we decided that we would like to have a distally mounted strain gauge which decouples the stress measurement from the optical design. So we used a single point load cell force transducer, which is pictured on the right here, where you have 4 strain gauges, which are essentially some thin wires in a grid-like pattern. So as you shear the beam, you're going to get sort of shorter or sort of compression or tension on the wires, which changes the resistance. And these are in a Wheatstone bridge configuration where you have 2 parallel arms. So therefore, as you apply a load, you can measure a signal across V out here. So calibrating this with known applied loads, we can create a calibration curve of the measured signal as a function of the stress that is applied to the load cell using the probe geometry. The results was we have a calibration constant of 410 kilopascals of stress per millivolt of volt of measured signal. The dynamic range of the load cell was 217 kilopascals and a sensitivity of 65 pascals. Integrating all these components together, so we have the optical probe, the load cell force transducer, as well as the handheld probe. So integrating these required no complex equipment and was designed as such, and also allowed for rapid interchangeability. So if we have a load cell that is malfunctioning, we can easily swap that out for a new load cell without affecting the probe performance. The assembly took less than 5 minutes and is calibration independent. So if you calibrate each section independently, you can combine them to an overall probe. So here's me putting the optical fiber into the probe, as well as a load cell, before bolting that together to an overall probe. So this is for the clear version, respectively. So in order to validate the probe, we needed to have some tissue mimicking phantoms. So I had an idea of creating phantoms that simulated a wide range of tissue elasticity, and I created 4 homogeneous phantoms as well as 1 bilayer phantom. So all phantoms were manufactured to be greater than 3 millimeters thick and 12.16 millimeters in diameter, and we embedded titanium dioxide scatterers to provide optical contrast during imaging. And as you can see from the graph on the right, we have the 4 different phantoms using different elastomers and mixing ratios that have different stress-strain responses and therefore different elasticities. For the bilayer phantom, I combined 2 different elastomers, of phantom 2 and phantom 3. So I had a layer of the phantom 2 elastomer and a layer of phantom 3 elastomer to create a bilayer material of the two phantoms, which are cured on top of each other. So going into the scanning protocol of how the probe is used. So for this proof of demonstration, we used a mounted setup so that we could ensure sort of accurate placement of the probe as well as the beam scanning parameters. So what we have is we have the probe, which is mounted onto an optical post, as well as a motorized translation stage. So we can finally control the relative position between the sample, which will be placed there, as well as the optical coherence probe. So we set some scanning parameters for the OCT, which I can explain in more detail if you'd like, as well as the load cell as well, and our load cell and electronics. So this protocol allowed for sort of a high sensitivity OCT to maximize the signal-to-noise ratio that we receive throughout the scan, as well as carefully controlling a full range of sample compression using the motorized translation stage. So in order to create sort of our elastography results, we needed to create some signal processing steps. So there are essentially two parts, which is our load cell processing as well as our OCT data. And combining both of these streams, we can generate a stress strain curve based on the stress applied from the load cell and the strain measured from the OCT data. All the processing was done using MATLAB R2016A. So for the load cell, the first step was to extract the raw data, which you could plot the signal as a function of the recorded time. You can see here that there is still a fair amount of, sort of Gaussian noise, which impedes accurate stress measurements. So in order to fix this, this is a subset of the data where we applied a median filter, which is a nonlinear filtering kernel of size 7, before also applying a moving average filter with corresponding to plus minus 40 milliseconds of temporal resolution. So you can see that it smoothed the Gaussian noise significantly through the scan. The next step was to crop the data into the corresponding region. So you can see here that there was no compression before 8 seconds, and then our compression stopped around this part. So we cropped the data to that region and applied our calibration factor 410 kilopascals per millivolt per volt that I described earlier, and therefore getting our measurement of stress as a function of time. Next is going into our OCT data. So what you can see here in the image is an axial intensity profile. So that's a one-dimensional intensity profile tissue as a function of time. So as you can see, that you've got your scatterers, which are compressing over the course of the scan. This was obtained from the complex valued signal. So in OCT data, we get a complex phasor as our result. However, for our M-mode image, we're only interested in the magnitude of that signal. So the way we can plot this is take our magnitude components by eliminating phase through this equation and converting it to a decibel scale for easier visualization. Next, we crop the M-mode data also to the region of compression such as the load cell, where there's no compression before this and after this region, and we also cropped it to 600 micrometers, our defined imaging depth for our OCT fiber probe. For the next step, instead of using the magnitude information, we're going to actually use a phase. So in OCT, the phase appears random, but is temporally invariant if there's no sample motion. However, if you do displace the sample, the scatterers create a new realization of phase, and therefore the phase shift can be related to the sample motion. So this image here is the phase shift image between consecutive A scans. And as you can see at the top, where at the interface between the probe and the sample, there's no motion as expected, within sort of increased motion the further down you go. However, here it is still quite noisy, so that we needed to increase the SNR. So this was done by blocking together 80 A scans and reducing the temporal resolution from 10 kilohertz to 125 hertz, which is the same as the load cell data. So this significantly increased the SNR, but you can still see some noise in the image. So we did some additional filtering steps of using a median filter down the columns, as well as a Gaussian kernel of 5 by 5 pixels to smooth the data. Taking our phase difference, this can be related to the displacement. given that we know the central wavelength of a system, as well as the average refractive index of our sample, we can convert our phase difference to axial displacement of our sample. So you can see this is agrees with our previous sort of phase difference image there. So we have very little displacement at the interface with the probe and then increased displacement. However, to go from displacement to strain, we take the axial gradient of the displacement with respect to depth. So this creates a strain map as follows. So as you can see at the start of the scan, there's large amounts of strain before you see stiffening of the tissue sample, which then you see lower amounts of strain through the rest of it. However, we want the cumulative strain that appears through the whole scan. So what we do is we take every column and we add them consecutively together to accumulate the strain. So you can see it starts with almost zero strain and then builds up through the rest of the scan to give us our overall strain in the sample. So to estimate the elasticity, we select a region of interest from my M-mode image. So here I've selected a 150 micrometer deep region in the central region of my M-mode image. And I take the stress and the accumulated strain that corresponds to this region of interest. To quantify the elasticity, it is common to have a 2D plot of stress versus strain. So we actually averaged the strain within this region and then produced a stress-strain plot corresponding to that region. So as you can see for the sample, there at low amounts of strain, there's low amounts of stress before it eventually sort of kicks up and becomes stiffer. So this is fairly typical of what you'd find for characterization of a biological sample. So after developing the signal processing steps, I demonstrated this probe on a few different tissue mimicking phantoms. So the region of interest were all 130 micrometers thick for all the phantoms, and the results were as follows. So this is for my soft phantom for the stress strain curve corresponding to our region of interest selected is as follows. So as you can see, to reach 50% strain, there's approximately 3 kilopascals of stress required. Going to the next stiffest phantom, it's still a very similar shape. However, instead of 3, there's now 14 kilopascals of stress, showing that it can differentiate between tissues of different elasticity. Doing this for all of the 4 phantoms, you can see that we start with our soft phantom and then go to our stiffer phantom sort of towards the end. So here it's clear that the probe can differentiate mechanical contrast between samples. We validated this against uniaxial compression, which is a gold standard in mechanical characterization. And you can see in the sort of later stages of strain, there is good agreement between the Quantitative Opticalcoherence Elastography probe as well as uniaxial compression. There is, however, significant deviation at low strains, and the reason for this can be explained by the different mechanical behavior of uniaxial compression as uniaxial indentation. Unfortunately, I don't have time to go into the full explanation. However, I will make myself available if you'd like to know more after the talk. So it is common to calculate the tangent modulus to provide one value to quantify tissue elasticity. So this graph here shows the tangent modulus to the stress-strain curves at 30% strain. So you can see there's a clear difference between the 4 different phantoms, validating the ability of the probe to do mechanical characterization. Looking also at the bilayer tissue, we had a top layer, which is 570 micrometers thick of a low scattering and soft material, followed by a bottom layer of a highly scattering and stiff material. So this is shown here in the M-mode image, where you can see the compression of both of the layers through the scans. Both of the regions of interest were 50 micrometers and corresponds to sort of small biological features, such as like a layer of tissue structure. So looking at the stress-strain curves for both of these regions, it is clear again that you can see the soft top layer as well as the stiffer top layer, and the ability for the probe to distinguish between mechanical contrast within a single sample in axial depth. Again, you can compare these to the uniaxial compression. However, it's important to note that uniaxial compression cannot provide depth-resolved mechanical characterization. So these curves are for the individual homogeneous phantoms. There is convergence between the two at higher strains due to mechanical coupling between the 2 different layers. Again, looking at the tangent modulus of our curves, we can see that the tangent modulus calculated 20% bulk strain for the probe, and the uniaxial compression is very close with about 10% error in difference. So a quick summing up the demonstration of the probe. So looking at the homogeneous phantoms, it showed that the quantitative probe had the ability to do inter-sample comparison between the different phantoms. This is important over the course of pregnancy, where you would expect the cervix to be stiff at the start of term and then get softer and softer. And this probe would allow you to look at the difference in mechanical characterization of cervical tissue as a function of time. As well, looking at the intrasample mechanical characterization that was done during the bilayer phantom. So not only could you look at the bulk mechanical properties of tissue, but also the remodeling of sort of intrasample layers such as the extracellular matrix or some sort of tissue structures within the sample. So conclusions. So over the last decade, preterm birth has become a significant problem and is becoming increasingly more prominent as the sort of preterm birth rate continues to rise. So this thesis has provided a demonstration of a proof of principle of an in vivo optical coherence elastography probe, and has been verified on tissue mimicking phantoms, showing the ability for intersample as well as intrasample mechanical characterization. So this provides the foundations for advancement to a truly handheld probe that can be further used to reduce the risk of preterm birth and the health consequences associated with it. Thank you very much, and I'd like to welcome any questions that you have. Who would like to go first?
Would you expect this method to be fairly resilient to patient movement? Would there be a large error that comes from that?
That is a good question. So the rate that the OCT data can acquire is at 10 kilohertz. So even sort of with the optical OCT, you can do measurements from 10 kilohertz all the way up to 100 kilohertz, and that is because there's rapid fluctuations in eye movement. So the OCT data is resilient to patient motion. However, the use of a load cell, that depends on how quickly you're able to read that sort of data. So if you have a load cell that you can have very quick and reliable measurements of stress, then it would be resilient to sample motion. But that depends on sort of the actual mechanisms that you've got within the probe. So this one, as a sort of a low-cost version, our load cell was $7 US. So we're kind of lacking in that electronic design. However, if you wanted to, you could scale it up to faster methods, which would be resilient to patient motion.
It's an excellent presentation. I like it. How far you are from clinically release your method?
I'm glad you asked that question. So future work. The first sort of look is we wanted to do a finite element analysis of how our probe, we can actually extract mechanical parameters, because we've used a compression model. However, you could see from the comparison with the compression model of uniaxial compression, there is some deviation there. So we'd like to do a finite element analysis, and I'd like to continue this research through a PhD to do quantitative optical coherence elastography scans of ex vivo tissue in a mounted setup, then doing in situ tissue scans in that mounted setup to demonstrate the ability to do real tissue scans. After that, we'd like to develop handheld scanning, which you'd need additional filtering algorithms to, do sort of patient motion as well as sort of hand motion from the clinician to compensate for that, before doing sort of clinical research using murine tissue, which is a fairly well-developed sort of path for developing new techniques, before looking for regulatory approval in human clinical trials. So to answer your question, there is still quite a lot of work, but however, we do have sort of a research plan of how this probe can develop to human trials. Yes?
Thank you, Kai. Great talk, and I like that it has like good application, and I see you've done a lot of work, which is good. My question was, when you tested the probe on the different phantoms, you chose the thickness of 130 micrometers. My question is, why, and is it clinically relevant?
So for this one, for here, so this region of interest, yeah. So the reason I chose it here is at the top of the sample, you get a lot of sort of tissue that's got high levels of friction. So we generally try avoid putting our sort of region of interest near the top. The reason I kind of chose 150 microns is that is like a general sort of thickness of tissue structure such as the epidermis. So the epidermis generally ranges between sort of 150 microns and 1 millimeter. So we wanted to demonstrate, could you tell the mechanical differences such as like the top epithelial layer of the cervix. So yeah, it was kind of informed by the tissue structures that we expect to get. Yep.
So we can have one question from Zoom. Any questions from Zoom? All good?
Can I ask one?
Sure.
That's okay.
It's great. Go ahead, Brandon.
Hi, great presentation. Do you think there could be any issue with heterogeneity of stiffness? So you've got a point measurement, so if you place the probe in different locations, you think you might get different measurements? Do you think that's something that
Yeah. Yeah, so that's definitely a point of interest. So during when they did elastography using ultrasound, that does give a wider area, and they showed good reproducibility between patients. So that's something that we need to check, is how reproducible is it for sort of heterogeneity of tissue. But yeah, being a point measurement, you are, without scanning mechanisms being utilized, yeah, you'd have to look at how heterogeneity is going to affect the sample, or even how much heterogeneity is within a single tissue, and therefore, like, what is your margin of error or your standard deviation of what you expect from one region to apply to the bulk tissue.
That's great. Thanks very much, Brian. Please join
Thank
me
you.
and